switch off the sound of YouTube, otherwise it will be complicated too.
Okay, we are on.
Just a minute.
You can start.
Switch off the sound. Okay, so good afternoon, everyone. Thank you very much for your attendance. Thank you for all the people that are following us on YouTube. Today, I had the pleasure to receive here in our course José Manuel Ugalde from the University of Bonn. José, I will introduce you based on the text that you provided us. So, José Manuel Ugalde is a biochemist from the Andrés Bello University since 2014. And he did his PhD at the Catholic University of Chile under the supervision of Dr. Loreto Joluigue, focused on the role of salicylic acid in the control of cellular redox homeostasis. During his PhD, he did 2 internships at the University of Bonn in Germany, in the laboratory of Dr. Andreas Meyer, where he focused on working with redox fluorescence biosensors. After finishing his PhD in 2018, he has continued his work on genetically encoded biosensors in Bonn as a postdoc, studying different aspects of redox biology in plants, focusing on different cellular subcompartments. Currently, he is developing his own research focused on the functional characterization of glutathione S transferases and their role in the redox control of plant cells. José is a member of the German Botanical Society, which awarded him in 2019 the Reinhold von Seebuch Prize in Dabrenhausen for his work on real-time monitoring of herbicide-induced photooxidative stress. And since last year, he's also a member of the Chilean Plant Biology Society. So, José, thank you very much for your kind acceptance for giving us a talk here. And the floor is yours.
Thank you. So, first of all, thanks Leandro, and thanks Igor for the invitation. It's quite a pleasure to be here. I wish I could be there, actually, but yeah, let's try and make the best of it. First question, can you hear me well then?
Yes.
Yeah, okay. So let me just share the screen. Okay. I don't know what you can see actually.
We can see your PowerPoint, but not in the presentation mode yet.
Okay, because I don't have, no, okay. Now
Yes.
It's presentation mode. Yeah, perfect.
Thank you.
Okay, so I will try to ignore the fact that we are live in YouTube and try to just make it as relaxed as possible. And is there any way that I can still see your faces? Yes, like that. Okay. Perfect. So, hi everyone, and thank you for joining us today. I would like to tell you a little bit about the work that we have been doing here in Bonn. And I call this this talk, I'm struggling with this stuff. Yeah, because I wanted to see your faces, and then the presentation, but I can only choose one, so I will go for the presentation. Okay, so I call it Let It Shine, the monitoring plant life dynamic with fluorescent biosensors. So, as Igor said, I did my training, my studies. I did it in Chile, so in the Universidad Andrés Bello, and also in the Universidad Católica. And since 2018, I have been a postdoc here in the plant signaling lab in the University of Bonn. Just for you to know, this is Bonn, a gorgeous city, very Germany with a river. We have like typical square with the monster and all that. And sadly, we're kind of here in the back. We're not right in the city center, but we're closer here to these towers in the back. But we have a nice forest next to us, so it's not that bad. And the city is popular for 2 things mainly, I would say. The fact that we have a lot of cherry blossoms, I think is one of the top 5 places in the world to see cherry blossoms. And because it's the place where Beethoven was born. So not that bad. Okay, but now with science. Fluorescent biosensors. Every time I start these, I think the best thing to start is just to describe then what is a biosensor. So by definition, a biosensor will be a self-contained integrated system that provides specific quantitative or semi-quantitative analytical information that consists of biological recognition element and a transducer. So an input and an element that translate in the information and then give us an output. And this should be in direct spatial contact. So I say this to the students, and of course it's very difficult for them to imagine this in a first glimpse. So the best way I had to show them this was just for them to imagine this. So this is Sting, the Stinger, the sword from the Lord of the Rings saga. And I don't know if you remember, but this sword, this little dagger, have a special quality, and is that the fact that it can detect when you have orcs around. And how is that? Because it can glow. And another example that I used to use with students is the fact, for example, of Harry Potter. So you all remember that when this bad guy is around, then Harry Potter have a sense of this input, and he will have his car pain, and he will know that Voldemort is around. And these are very good example of sensors. So we have an input and we have an element that do a transformation and deformation, and then we have an output. But well, the plant cell doesn't have orcs or magic, but it has, for example, reactive oxygen species like H2O2. And we don't have the dagger or the scar, but we have, for example, fluorescent biosensors, genetically encoded sensors. Like in this case, these cartoons of GFPs. Well, we have different colors and flavors of fluorescent biosensors, and this is a nice kind of overview of them. In this case, for example, sensors are, let's say, for measuring changes in hydrogen peroxide, or changing measurement or measuring changes in the glutathione redox potential in the cell. And they have in common that they use fluorescent proteins. I will not much go into detail in all of them. The ones that you need to know today are GRX1, ROGFP2, ROGFP2 ORP1, and HYPA. So these one have in common that we have operator. So we have this element that do the translation of the information, and we have an output, which will be the fluorescent protein. And for example, in the case of the ROGFP2 based sensors, we will have this protein that will have in here show these two cysteines that can form a disulfide bond only after this operator or this facilitator can sense initially hydrogen peroxide, then it will resolve this disulfide bond that will then be transferred to the protein, making a change in the fluorescent protein, in the fluorescent properties of this protein. Or another example is HYPA, where we have a CP-YFP, a circularly permutated YFP that can that has two domains that senses hydrogen peroxide, and these are domains from a transcription factor or in bacteria. And when they sense hydrogen peroxide, they will interact, forming a disulfide bond again that will change the spectral properties of the protein. Okay, but in detail, how this work? So the ROGFP, there is the protein or the sensor that we use the most here in the lab. It's engineered based on the normal GFP that you may already know. And what this sensor has is two extra cysteines on the surface of the beta barrel that are marked here. And these ones are quite close to the chromophore. So when the sensor is fully reduced, we will have a spectra of excitation of this protein that will have a peak around 480 nanometers, which is the normal peak of fluorescence of a GFP. But when this sensor encounters or senses a surrounding or oxidized, these cysteines can form a disulfide bond. It will create this oxidized thiol, and it will change the properties of this excitation, where now the peak at 480 will decrease, and we will have a new peak at 405 nanometers. The good thing of working with proteins, with genetically encoded sensors in this case, is that of course they're proteins, and we can put localization tags to them, so we can send them to different compartments as we need, like putting a zip code on their amino terminal sequences. And with this, we can, for example, target them to plastids or to the cytosol or to the mitochondria. So it allows us to measure specifically in these different compartments the whatever it's measuring. And in this case, for GFP2 fused to GRX1, it will be measuring the glutathione redox potential. And these sensors, when they are in these compartments, their properties are very much alike as the purified sensor. So you can compare the excitation spectra here compared to the purified, and they're quite the same. And this is true for both the GRX1 RGFP2 and the RGFP2 fused to ORP1, which is a hydrogen peroxide sensor. Okay, so I'm not sure of if you have seen papers on the use of these biosensors, but in the past, let's say like 2 years ago, the main way that we have to image these sensors, and therefore the changes in real time in the cell, was using confocal microscopy. And this is great to have a very nice resolution and spatial resolution, like to see in the cell actually what is happening. You put your sample in a confocal microscopy, in a CLSM, and then you can image these two channels, the two peaks of excitation of the RGFP, 405 nanometers for the peak of the fully oxidized form, and 480 nanometers for the peak of the fully reused form. And you can make a ratio of the intensities of these two channels. So then we can make a false coloring image of this ratio, where the lower the ratio is of this channel, the 405 over 480, the lower it is, more reduced will be the cell. And we can put them a false coloring here from blue to red. And this is all great. We can really see in one single cell what's happening. The problem here is that we can see then only a group of cells. We can see only this happening for one single plant at a time, and not for very long. You cannot have a plant in the CLSM on a slide, let's say for like 2 hours or so, unless you have a special system for that. So what we have been doing here is to switch this to a plate reader fluorometry. So we use a fluorescence plate reader, the ones that you can use, for example, to measure I don't know, cell cultures or purified proteins or whatsoever. And this one, it's equipped with the filters for the RGFP. And what we can do is, instead of having one plant, we can pool them. We can put in one single well, in this case, 3 to 4 or 5 seedlings. And if we put this in an fluorescent loop, you can see clearly here that the GFP signal is quite bright. And then for each one of these wells, and let's say that we can have at the same time 96 wells filled with plates, so we can have a lot of sampling. We can use the filters to excite close to the peak of the fully oxidized, the peak of the fully reused, and then collecting a 520, making the ratio. So I make this sort of introduction for the methodology because it's pretty much the same that we will be using for all the work we do with the sensors. And the results look something like this. So we plot the ratio over time. And remember that the ratios, the higher they are, more oxidized is the sensor. And we can calibrate this with DTT, with hydrogen peroxide, or with other oxidants, and we can have a dynamic range between the fully reduced and the fully oxidized form of the sensor. So when we put control plants, so plants without any treatment, the sensors are fully reduced, so they are equilibrated with the glutathione pool, which is highly reduced in cell. But then we can play with this. We can force oxidation of the cytosol, and by that we can use external oxidants, for example, hydrogen peroxide. And the cool thing is that we add different concentrations of hydrogen peroxide, and we can oxidize in a very fast way the cytosolic glutathione redox potential, and respond very fast. And also we can see after time now a recovery. Let's say after 6 hours, we can go back almost to the initial redox state after 10 millimolar adding of hydrogen peroxide. And this is also true, for example, for the sensor for hydrogen peroxide, ROGP2 or 1. So we have a very fast and transitory oxidation of the cytosol. Now, the cool thing is that with this setup, we can test different things at the same time. And in this case, we were checking, for example, how this detoxification will occur in plants that lack, for example, catalase, right? So the enzyme that degrades hydrogen peroxide to water in the peroxisome. So the more active one in plants here is CAT2, and in Arabidopsis we use our sensor in this mutant background. We fed the plants with hydrogen peroxide. We have an oxidation, but then we don't have a reduction over time, which indicates that these plants are indeed impaired in the detoxification of hydrogen peroxide. And this is for both sensors. If we analyze glutathione redox potential, or if we analyze only hydrogen peroxide amounts in the cell. And here you can see the controls of how these channels behave radiometrically, and this is very important. These are sensors that they're not intensiometric, so they don't. We don't need like a second point for calibration. These are self-calibrated. If one channel goes up, the other one will go down. Okay, so we can test this with externally added oxidants. But now, what happens if we want to create an oxidative stress within the cell without applying any external, in this case, ROS directly? And for that, we switch for a model. We switch for herbicides that induce oxidative damage in the cell by the hijacking electrons from different parts. So in this case, we use methylbiologen, which is a very toxic herbicide. It's non-selective. It kills pretty much everything green. And after several years using it in the fields, people realized that it's not only killing the green stuff, but it was only very dangerous for the farmers. So they started to develop Parkinson and other diseases, the degenerative diseases. In the plant, this methylbiologen will induce the collection of genes that are related to oxidative stress, for example, the transcription factors at 12. And how this works? Well, in plants, methylbiologen will hijack or redirect electrons from the photosystem 1 to molecular oxygen. And we will start creating a superoxide that will then be dismutated to hydrogen peroxide. But remember that it's also very toxic for the farmers. So this methylbiologen can also redirect electrons from complex 1 in the electron transport chain, creating also superoxide and finally hydrogen peroxide, which, if cannot be controlled, it will just start creating oxidative damage in the cell and also affecting the glutathione redox state, which we can measure with GRX1 RGP2, or we can measure the amount of the changes in the dynamics of hydrogen peroxide with RGP2 or 1. Okay, so same setup, but instead of hydrogen peroxide, we put methylbiologen, enhancing the production of hydrogen peroxide via the plastids. So we see the cytosol, and the oxidation was not that impressive. So we have a small shift, but pretty much not what we imagine. But in plastids, we see that this oxidation is quite more pronounced, which at the end makes sense because it's the place where the hydrogen peroxide is being produced. Also, mitochondria, we see a change, but not compared to what we saw in plastids. So if this is produced in plastids, then it's fair to think that if we enhance or if we activate further the electron transport chain in the plastids, we may have more ROS. And in plants, it's quite easy to do. We can just put light into the system. And this is exactly what we did. So we fed the plants with methylbiologen, we leave them for a couple of hours, and then we give them a pulse of light. And you see that, for example, here in the cytosol, now we have a jump on the oxidation of the sensor, indicating that we can indeed boost the oxidation of the sensor. We can create photooxidative stress, and we can follow it in real time in the cell, in different subcompartments of the cell. So for the end of this first part, this is all already published, and you can check it. We managed to have the sensors for the glutathione redox potential and for hydrogen peroxide expressed in different subcompartments of the cell. And then if we add illumination in the presence of methylbiologen, we can produce a photooxidative stress that not only will oxidize the chloroplast, but will create enough ROS, it will create enough oxidative stress that will affect the other subcompartments next to it. Yet, as a point for connecting to the next part, we saw that if we put this sensor in the cytosol, we still don't have an oxidation that was happening also on the background. So it's always the question then, okay, we have sensors, they're working, but can we also push forward to create more sensitive sensors? And this is something that we have been also working on, and I'm kind of shifting a bit faster to don't spend too much time. And this is a work that also we were doing last year, especially, and it's with new genetically encoded sensors. So in this case, have Hyper 7. This is of course, the 7th iteration of this sensor. And as I mentioned before, this sensor, it's a circularly permutated YFP that has two, let's say, domains of the domain that senses hydrogen peroxide from the OxyR transcription factor. So you will have a part that binds to the DNA and another part that will sense hydrogen peroxide. And this sensor only have the part that senses the hydrogen peroxide. So when it senses it, it will create this disulfide bond that will change the properties of the Hyper 7. So it will change the spectral properties of the CPYFP. And all of this together is called Hyper 7. And we have this already published in PhD, and we were very lucky that our artwork was also selected to be the cover of the journal that month. And I also use it for the poster for today. But going into it, and this is kind of the pipeline when you develop a new sensor, we express it in plants, in this case in Arabidopsis, and we tested that we can image the different channels. In this case, for the Hyper, it works a little bit different. So the oxidize or the peak of fluorescence in the oxidized form is at 480 and the reduced is 105. And the thing that you want to see is that when you express your sensor in plants, you don't create, for example, an effect or an impaired growth of the plant whatsoever. So we proved that the plants grow just normally. We didn't saw any obvious phenotypes on Arabidopsis plants. And then we analyzed their spectra, same as we can do for the ROGFP. And here you can see that in vitro, we can purify the sensor, and the sensor can be almost fully oxidized with 0.1 millimolar of hydrogen peroxide, or 10 millimolar hydrogen peroxide is indifferent. So we have a very sensitive sensor. and we can fully reduce it with TSEP, which is a strong reductant, but not able to cross membranes, so we cannot use it on plants. But sadly, with DTT, nothing happened. So we have a sensor that, even if it's very sensitive to hydrogen peroxide, we don't have a way to reduce it. And in vivo, of course, the sensor reacts to hydrogen peroxide, but in the cell, apparently, it's we believe that it's fully reduced. Again, we cannot know since it doesn't respond to DDT. Which match very much to the reported spectra of hyper 7 already reported for the purified protein. Now, one of the particularities of the ROGFP2 based sensors is the fact that they depend on the glutathione redox potential, of course. So if we grow the plants on BSO, which is an inhibitor of the biosynthesis route of glutathione, we will create plants that do not develop. And these plants, when we analyze the redox state of the ROGFP2, and this again, more red is more oxidized, more blue is more reduced, we will see that the sensor is fully oxidized when we work with ROGFP2 based sensors. But we saw that Hyper 7, same as the previous version, Hyper is not oxidized under BSO treatment. So we grabbed these small plants, we imaged them, and the sensor was not oxidized, which tell us that it's independent of GSH, which is very good for example, working on different biological questions that we may have in the future. And other interesting fact is that when we put hydrogen peroxide on these plants and we image the sensors, we saw that the ROGFP2 respond as expected. We have a fully oxidized sensor on the hydrogen peroxide treatment. But and also we saw something similar to Hyper 7, but the whole Hyper, we saw this oxidation only on leaves. We didn't saw it on the root elongations or the root tip. And then we ask ourselves why, if we are putting hydrogen peroxide, we will not see an effect on the old Hyper. And we believed that it was due to a change in the pH. Because if you add hydrogen peroxide, you will have an acidification on the cell. And this we proved by just using CPYFP that alone works as a pH sensor. And as you can see here, if we add hydrogen peroxide, we have a shift on the ratio of this sensor. And this sensor, as I mentioned before, it's pH sensitive. So the old Hyper, it's depending on pH and is not responding as well as the Hyper 7 due to this fact that it's very pH sensitive. Now, when we clamp the cytosolic pH in the root, you see this artifact that is created on the old Hyper, when at higher pHs or lower pHs, we have a change on the dynamic range of it, while Hyper 7 is quite stable. Okay. Now, knowing the kind of like the introduction to this Hyper 7 in plants, we now wanted to know the most interesting fact of it. Okay, as I mentioned before, is this a more sensitive sensor? And for that, we use this system. It looks sort of complicated, but it's just a perfusion system on attached to a CLSM, where we can put a seedling in this chamber here, and we can perfuse treatments throughout the seedling in a continuous way, and these treatments can change as you want. And for example, in this when we set up these, and this is just this week published in Methods in Molecular Biology, we can pump hydrogen peroxide through the seedling, and the ratio, meaning the oxidation of the cytosol in this case, will be followed in real time in this in this matter. So we can oxidize the sensor, in this case, both the sensor for hydrogen peroxide and for the glutathione redox potential, and we can also reduce it. So we have a sensor that it's dynamic. It doesn't remain fully oxidized. And here I put the QR if you want to check the preprint. So we use the same setup, and we went back to the Hyper 7 and give them first to the plants a little bit of buffer, and then we perfuse different concentrations of hydrogen peroxide and then buffer into DT. So what we saw is that the all sensors respond, but they respond in a different way. So when we put this in plots, when we pipette or perfuse 1 millimolar hydrogen peroxide, all the sensor responds. So here's a part of the pumping of hydrogen peroxide, and we will have an increase of the ratio indicating an oxidation, where Hyper 7 was a little bit higher than ORP 1, which is the previous version of hydrogen peroxide sensing sensors. But when we use very low concentrations of hydrogen peroxide, in this case 10 micromolar of hydrogen peroxide, very low, we saw that only Hyper respond. So we have indeed a very sensitive sensor for hydrogen peroxide, which was very nice for us. But this in roots. And then I was just imaging different aspects of the plant or in different conditions and so on. And while I was imaging leaf cells, I realized that if I make different zooms of the cell, the sensor started to oxidize in real time. It was quite puzzling and quite frustrating as well. And here you see that on different zooms. So the left is less zoom, the right is more zoom on the cell. And this is just a merge. This is not this radiometric image when we have false coloring, but only the merge. And as more green, it's getting more oxidized. So I make the radiometric images of these, and we saw the same. So the more zoom we have, so if we have the same amount of lasers now in a smaller surface of cells, we will have higher oxidation over time. And this is not much time, it's just 2 minutes of exposition to the lasers. So we thought, like, okay, why is this? Is this the sensor itself, or this is something that the laser are causing on the plants? And we tested the other sensors as well. And you can see here on the top, so only Hyper 7 is changing over time. The old Hyper is not changing. And in the case of the raw GFP, where the peaks of the excitation are different. If it will get oxidized, it will be more magenta, which is not. We saw that only Hyper is responding this way over time. And not even the purified protein is responding like that. So it must be something in the cell that is causing this oxidation. And the most or the more obvious candidate here will be photooxidative stress, right? So we are putting a very energetic light that is creating rust via the chloroplast, and that rust is sufficient to oxidize the sensor. And we tested that by putting side-by-side plants with and without DCMU. And DCMU is also an herbicide that inhibits the electron transport chain in plastids. And the result is quite clear. So if we put DCMU, the cells do not get oxidized over time with this laser. So it's indeed due to the photooxidative damage that we are creating. And the good thing is that this is also reversible. So if you have a zoom and then you give them 2 minutes of intensive laser imaging, you will have an oxidation of the sensor, and then if you leave it over time, it will recover, and then you can oxidize it again and again. So we have a sensor that is dynamic. And it opens a very interesting question, because when we image, for example, mitochondria or smaller organelles, when you make a big zoom out of it, you use these sort of laser powers and laser concentrations in this small area. So we just now have the sensor to tell us that we are creating ROS, a ROS that we were not able to measure before in plant cells. So imaging itself, it's a stress that we were not considering, at least in a way that we can measure. So that was quite interesting. And we tested the other stresses that I mentioned before. So for example, methylhalogen, and now in the cytosol, it's quite clear that we have an oxidation that we didn't saw with ORP1 or very, very mildly, and then with hyper 7, it just is much more increased. And not only that, but if we, for example, measure the ROS created by an elicitol like flagellin, we can see that the response is much better. So as a summary so far, I can tell you that we have adapted this hyper 7 sensor now to plants and allow us to have a very sensitive sensor for hydrogen peroxide that can sense as little as 10 micromolar of peroxide being added to it. And it's so sensitive that also respond to even to the illumination that we use to measure the sensor, and allow us to follow different biological processes that induce ROS production, such as flagellin or methylhalogen. So that's the end of the second part of us creating a more sensitive sensor. But at the same time, different compartments of the cell will have different requirements for sensing. So for example, this hyper is very good for cytosol, but I don't know if we put it in the plastic, maybe it will be fully oxidized or not. And depends very much on the context of each subcompartment. For example, which sensor we can use and which questions we can answer. So the last part that I will mention in this talk, it's about a little bit of the work we have done on dissecting key components for redox homeostasis, but in a specific compartment. In this case, is the endoplasmic reticulum. And this is the preprint, so you can also scan it and check it later if you want. And we focus on trying to dissect key components of the maintenance of the redox homeostasis in the ER, in this case, the endoplasmic reticulum oxyreductins. And we're very happy because this paper just got accepted last week, so it's a pleasure to share it with you. So we talked already about ROGFP, and we talked about the fact that we can put it in different subcompartments because we can put signals and we can direct it to signal peptides, and we can direct it to different subcompartments. And we have done that, and it has been done. So for example, here we have a peroxisomal GFP or a chloroplastic GFP or ROGFP targeted to the ER. And the difference is quite clear on the oxidation. So if we see this bar in the right, while other compartments remain fairly reduced, if we put the ROGFP in the ER, it will be fully oxidized. So different to other subcompartments, the ER has a more oxidative environment. And it makes sense when we think, for example, the function or the main function of the ER, which is oxidative protein folding. So what we have here is a subcompartment that will be fed on these nascent peptides that are entering to the ER, and they need to be correctly folded. And by that, they need sort of an oxidative power. And this function of the ER, I like to imagine it in my head as sort of as an origami. So a protein, before it's mature, before it's folded, or before it's, for example, forming dimers or a maturation or active sites or other examples, it's like a piece of paper. It has a lot of potential. But then when it's folded and it's active, then we have, for example, a crane or a boat or whatever you want to do. So these nascent peptides need to get oxidized, and for that we need to transfer the electrons from these cysteines marked here with these gray boxes. And this will be this transfer will be facilitated with protein disulfide isomerases in the cell, in this case in DR, and these electrons will be then transported from the PDIs to the to eros. So this oxidized PDI will then get reduced by accepting these electrons from the nascent peptides that will get oxidized, and then these electrons will be transferred to eros, which is the ER thiol oxidases in the ER. And finally, these electrons need to go somewhere, and the final acceptor of electrons will be oxygen that will create then hydrogen peroxide. So we ask ourselves, how then will a plant do, and how the glutathione redox potential of the ER will change if we take eros? And in the case of Arabidopsis, it only has two. So we believe that they are very important in the plant, and we were completely right, because if you take both eros at the same time, then the mutant is lethal. The plant just lose all the will of living, and you will never get a double mutant. And this was already published a couple of years ago. The most you can get is the heterozygous and homozygous for the other, and you will already see these empty spaces indicating embryo lethality. So we did the best next thing, which was to create or to express an artificial microRNA against ERO2 in the mutant background of ERO1 knockout mutant. And we created a line that is completely knockout for ERO1 and has reduced expression for ERO2. And this plant, this line 5, it doesn't have a clear phenotype in normal conditions. It's viable, it exists, it can grow, it has its full life, doesn't have major phenotypes. But then if we put these plants into a reductive stress, such as DTT, we saw that the plants are highly sensitive to it. So with 450 micromolar, you can clearly see here that the double mutant is not having a good time. And this phenotype occurs as at very low concentration, as little as 200 micromolar of DDT. We already have these impaired primary root growth. So this double mutant is very, very sensitive to reductive stress. But now if we want to measure this, of course, we want to use our sensors. So we know, when I told them already, that the redox potential of glutathione in the ER is less negative than the cytosol. The cytosol is if we put a ROGFP, it will be fully reduced, which is close to minus 310, and the ER it's still debatable between minus 208 or 220, depending on the paper you read. So here I put this table to make it more understandable. In the bottom we have the cytosol, in the top we have the ER, and then we have a table of the redox potentials. And ROGFP2 has a midpoint potential of minus 280 millivolts. So when we image the cytosol, it just fits to have it fully reduced. But anything, let's say, on top of minus 240 millivolts, it will appear fully oxidized for the sensor. So we cannot resolve anything. It will just always be fully oxidized. So therefore, for example, if you see these, you have the ROGFP2 targeted to the cytosol, and these are guard cells, and in the merge they look like this. And if you target the same sensor now to the to the ER using the skitinase target peptide and a retention signal, it will look quite different. So both channels, you see how one goes up, one goes down. So this sensor is just not good for imaging into the ER. So therefore, we did a new sensor, which is called ROGFP2IL, that has less negative midpoint potential and allow us to image now in a dynamic way the ER. And we express this in the mutants that we saw before, so in wild-type plants and in the single mutants and in the double mutant for EROS. And in a second. So now the raw GFP is not fully oxidized, but it's somewhere here in the middle. It looks kind of greenish. So we can measure differences high and low. But the most striking thing is that if you see the sensor expressed now in the ERO1, ERO2 double mutant, is less oxidized than in the wild type. So if we see this in numbers, so that for wild type, we can extrapolate that the midpoint potential here is around 240 millivolts, but for the ERO double mutant, it's more negative. So it's indicating that it's more reduced. So at minus 253. And we wanted to measure, okay, if this is more reduced, and we know that it's plants that are more susceptible to DTT, how will be the capacity of this plant to survive and to overcome reductive stress? And this is something we have also put some effort into it in this previous plant cell paper. This is the same setup we wanted to use, where we have our sensor in the ER. We can put some DTT, and then we can evaluate how much it takes for the plant to recover. And we did this with our wild type and with our mutant, and analyze after, for example, the reduction phase, how much it takes for going back to the basal state, to the reoxidation. And both the rate of reduction was similar to both, and this is the same perfusion system that you saw before, but the rate of reoxidation was slower for ERO1, ERO2. So these plants, beside having a less oxidized environment, has more problems maintaining the redox homeostasis under reductive stress. And the final part of it was to ask, okay, we took out EROs and we have plants that are having a difference, having an impact direct that we can measure in the glutathione redox potential of the ER. Now, what would happen if at the same time we take out the final electron acceptor, if we take down oxygen? And that is something relatively simple that we can do and just expose these plants to hypoxia. So we did that. And the plants here are the wild types. And the hypoxia, you have some sort of damage, some wilted, some plants that will have these more dead leaves. And if we quantify this in both the wild type and the ERO double mutant, we see that the percentage of dead plants are, or the sort of overall survival score in these plants, it's lower than the wild type. So we have plants that are more susceptible to hypoxia or to low oxygen conditions. And this is not only for seedlings in plates. Here in the top, we also did submergence. So these were plants grown on pots that were submerged in water for 4 days or 5 days, and then the fresh weight of the rosette was measured, and indeed the double mutants are more affected. Finally, here we also measure the ability or the capacity of these mutants and the wild types to regrowth the root tips after hypoxia, and again, they are more affected. So this is the phenotype in the macro, but what will happen with the redox potential now of these plants under hypoxia? And in the plate reader, that was challenging to do, but not impossible. We use a system that has attached to it an atmospheric control unit. So a machine that allows us to control the amount of oxygen inside the system by pumping nitrogen and pushing outside oxygen. And we tested first different regimes of hypoxia. So, for example, short 1 hour, 2.5, or more hours of hypoxia. And the thing you can see first is that at all points on the onset of hypoxia, we have a reduction of the redox potential, or we have a reduction of the oxidation level of the sensor in the ER, followed by coming back to the normal homeostasis, to the normal levels that has at the beginning. And when we pump oxygen back into the system, we will have a burst of oxygen, reactive oxygen species. We will have an oxidation of the ER. So we compare this is only wild type plants. So this is kind of the signature curve we will have on the hypoxia, and we compare it with the mutant. And the single mutants look very much alike, wild type, but the double mutant has a very particular trend where it's very much impaired to go back to the normal homeostasis how it started, pretty much according to what we saw before. So if we take out the eros and if we take out the final acceptor of electrons, we will impair the normal homeostasis of the glutathione redox potential in the ER. Okay, so far I can tell you that we created this double mutant that is very sensitive to oxidative stress, to reductive stress. We have phenotypes up to as little as 200 micromolar DTT, that these plants have a more reduced ER, which is of course not good for the plant, and that is also affected under hypoxia. And we're still kind of evaluating and thinking on a model on how we can explain this, especially because that we don't have this oxygen as a final acceptor for electrons. So now the question is still on, like, who is the final acceptor of electrons where oxygen is not there? So we're still working on it. But of course, the interesting thing is that under hypoxia, we will at the same time lose the reductive input of the newly synthesized peptides into the ER. So this will, on hypoxia, this will be reduced, and this will also affect the normal redox homeostasis of the ER. Okay, and we're a little bit over time, but as a final thing, I just want to tell you like the outlook of new questions that we have for the future. For example, of course, we have this transport of ROS, but how is this ROS being transported between these different compartments, or from the outside of the cell to this to the cytosol? Like, which are the key I don't know, aquaporins, for example, they could transport this hydrogen peroxide, or at the same time. We know that the peptides are transported, newly synthesized peptides are transported into the ER via 661 translocorn. They are matured and so on. But at the same time, we have other components in the ER like GRXs. Now, we want to elucidate, okay, what could be the role of these GRXs into maybe oxidative protein folding or not, or GPXs as well in the ER. And finally, if we have these sensors now, of course, everything in the model plan of redoxis, how could we continue this research, but now with more global of crops of global interest, such as rice or tomato, barley, and this is already in going. They're more or less success on creating these plants, but we're very much enthusiastic for the future and see what questions, what answers we can get evaluating these in these plants. And just as a final thing, to thank the lab. It's a great place to work. We have very motivated people, very, very talented people, and I'm very grateful for being here. And and also to the collaborators we have in different parts. Yeah. And also, I think Thomas should be around, and it was also a pleasure to have him from Brazil. So with that, I think I will just thank you all, and I will just stop sharing now, or maybe you would just leave it like that.
Okay.
I'm very sorry because actually I couldn't see any faces. I was presenting to just my.
This is a drawback of online presentations in the pandemics. You were like talking alone. You don't see any faces. Whereas you're watching.
I couldn't see even the faces. Yeah.
Okay, so thank you very much, Jose. It was a very interesting talk. So I will open for questions. People there and on YouTube, I would like to ask you to let your questions in the chat that we can read this to Jose. So maybe I will start, Jose, with a question until the students will warm up with their own questions. Most of the work that you presented, I guess that you worked with Arabidopsis seedlings, right? I was wondering about the possibility, if you want to check, I mean, of course, that you're treating plants with hydroxide peroxide and some compounds, but what about if you want to study this on fully grown plants? Is there any drawback? I mean, can you take like a piece of a mature leaf or you know, this type of maybe a biological process that is not relevant in the seedlings but is relevant in fully grown plants? Is it possible?
So yes, and it's a key question because, for example, not all physiological processes will be equally active during the life of a plant. So just like, for example, the FLS2 receptor for flagellin, right? You need a certain point of the plant life to have it to make this function. So I will let me hide this and so short story. You can for example put leaf discs on it. You can cut pieces of the leaf. You can put it on a buffer, and you can evaluate it. There are plenty of work now as well using more sophisticated hardware where you can image full plants, and I can maybe show you, yeah, something like this. Let me just close this part. This is a system. It's Iris Lumina and also Shiloh Rosenwasser have been working with with system like this, and also Ron Mittler in the US, where you can image a full plant, and not only a full plant, but you can image like in this case a full pot with 3 plants, and you can just spray them with different treatments and follow the redox rate on a temporal resolution as well, but with full grown plants. So it can be done, right? But you need to have the money to buy this machine because we only have it for testing, and it has a lot of potential. But yet again,
yeah, because I mean, you show like in your one of your last slides that you're moving towards crops, and I guess that it would be much better to analyze this type of things when the crop is fully grown. I mean, we're mainly not interested in seedlings of the crops, right?
Exactly. So I think you're limited to the space of the camera, of the limit to the space of the chamber, sorry. And if you can put it there inside and just focus it, you can image it.
Perfect. We have a question on YouTube from Professor Marcia Margis from Brazil. She congratulates you for your beautiful work, and she asks which of these sensors have already been tested on monocots, such as rice, for example.
So I will say at the moment we are trying to put it in rice, and this is a collaboration we have with I put it here at the very end. Wait, sorry, I have it. So yeah, Chen-Lin is trying to do this. He was working here in Germany, and he's trying back there in China to put the raw GFP into rice. Yet we don't have any successful transformants, unfortunately.
But there is a problem in the transformation, or
I don't know. Yeah, unfortunately, I'm pretty much focused on Arabidopsis at the moment. So I depend on his ability to transform it to make it work.
you mentioned that in Arabidopsis, I mean, I guess the sensors are expressed with a constitutive promoter.
Yes.
And you show that there's no yield penalty. I mean, there's no growth problems for Arabidopsis.
No, but for example, we have seen over time that, or time and time again, actually, that when you express, for example, sensors in the mitochondria, we do have an effect of growth. So the plants grow smaller. So the tool is also the constitutive expression of these sensors, depending where it can cause detrimental effects on the growth of plants.
And when this happens, I guess that you cannot use, I mean, because then you cannot untangle what is the result of, I don't know, the expression of the sensor itself.
I would say that you can use them, but very carefully and very honestly, right? So if you are comparing, for example, certain components of the electron transport chain, if you're evaluating different mutant backgrounds and the phenotype is comparable between them, you can, but yet again, you need to be just cautious and very transparent about it.
Yeah. Yeah. Okay. Let me check if there are more questions there. No, just people congratulating you. Flavia, yes, you can speak.
Thank you. I would like to thank you for the interesting lecture. And I have a question. It's more of a curiosity. Do you have an idea why the Hyper 7 is more sensitive than the Hyper sensor, biosensor?
So this was pretty much, thank you for your question. This was pretty much a tour de force. So this was developed by Valery Pak and Seva Velusov. And what they did was a number of mutations on the CPYFP to make it pH insensitive and to make it more sensitive to hydrogen peroxide. And also what they did was a screening, and I think I can put back that slide. So they tested a different set of these oxy-R domains from different bacteria. And at the end, they found out that the one from Neisseria meningitidis was the more sensitive one. So they just collected or purified a clonal number of these oxy-R domains and just tested one by one. And they find out that this was the most sensitive one. So in the reality of this Neisseria meningitidis, apparently it has the ability to sense better hydrogen peroxide. So it's just trial and error until they found it.
Okay, I understand now. I thought there was something about the structure, but they just did a screening, right?
It was a screening, correct. But they
Okay.
have made different variations on the protein to make it more stable, to alter the surface of the protein. But I think the key point here was to test different oxy-R domains.
Okay, thank you.
Jose, we have more questions on YouTube. Ana Luisa is asking you, would it be possible to use or monitor those sensors, especially Hyper 7, in different developmental stages, like seedlings, like seeds, first minutes of germination, for example?
Yeah, and there is a very, very cool paper from Thomas Nitzel where they followed the redox changes of seeds upon embedding, like when you water them. And remember that seeds can stay quiescent for, I don't know what's the record, like hundreds of years or whatever, this one in Jerusalem. And all of a sudden you put them water and they have this switch, this very a strong switch of metabolism, and that is also redox changes. And you can see that on seeds, and it's very fast upon water exposition.
She mentions that paper of Nitzel, 2020, and she asks if there is any difficulty using seeds. I mean, compared with
of course, yes, of course. So similar to using pollen, if you want to do this as well, you have an amount of autofluorescence that you need to take care of. So the seed coat is also quite autofluorescent. So the thing we do, and it's the recommended thing to do with any work with biosensors, is to use the same tissue but without the sensor, and we use that for autofluorescence subtraction. So you put your seeds wild type, then your seeds with the sensor, you make the same experiment, and then you subtract the autofluorescence, and then you should get only the or hopefully only the signal from the sensor. And of course, you depend on the strength of your sensor. Some sensors might be more bright than others, and you always want to have a good signal to noise ratio very high. Like you want more signal, you don't want too much background.
Okay, thank you. I think she has a personal interest in seeds, so that's why she asked you. I also have one question. It was interesting when you said that your sensor, upon exposition to the laser, was like oxidizing. And I was wondering whether when you're using, I don't know, confocal microscopy with the laser intensity, if this is not creating somehow an artifact. I mean, let's say that the laser intensity would create more reactive oxygen species, for example, and then that could be somehow.
That's the point. That's the full point. So I think we tested it here. So what I was thinking is exactly as you say. So you put a laser, and then that, in my idea, was creating more ROS in the plastids, because now the plastids have too much, a very large stimulus. You are really pumping photons there. And I thought, okay, if I do this now with an inhibitor of the electron transport chain here, I might stop the oxidation, and it's exactly what we saw. And furthermore, I'm sorry I didn't put it here, but I also did the experiment with only one wavelength. So for example, I only excited with 405 until the end, and then make the imaging, and then only with 480, and then the imaging at the end. And the 405 channel, it's the main responsible of having this oxidation. So now a next outlook, and it's something that might or might not work, is to fuse or to create a hyper that do not depend on 405, because it's a very energetic, or it's not very, but more energetic than 480. Yeah. Remember, like the shorter the wavelengths, more energy we have.
Yeah.
So for example, you have another protein, let's say a RFP, a red fluorescent protein, or something that we can use to make the radiometry. Maybe we can avoid to use the 405, and we can avoid this artifact. But yet it's something that we have been doing for so many years, and now we have this sensor, and it's telling us, okay, something was happening that we were not seeing before.
Okay, perfect. Any more questions? I don't think we have more questions on YouTube. Students? Well, I think that's it, Jose. I would like to thank you very much once again for the very interesting talk. It was a pleasure to have you here. Maybe in the future we can invite you to be in person, like you said. And thank you very much for sharing a little bit of your work. Nice that you have a lot of publications like going out now and to be published as preprint now. So thank you again on the behalf of everybody from this course, Leandro, myself, Magda, who was not here, and the students. I would like to thank you for your contribution.
Yeah, just again, thank you for the invitation. It was great. I'm very sorry that I might have gone maybe a little bit fast, but I didn't have any inputs from it was just the presentation. I couldn't see any faces or whatsoever. But you can put you can change the time on YouTube and play it slower, so that's a cool thing.
Yeah.
And also just an open message to everybody. If you have any question or any doubt in these days or whatever, I just want to speak about sensing or new ideas. Just feel very free to just write me an email or just send me a message in Twitter. Yeah, sometimes sometimes I put funny stuff there.
Okay, thank you very much. Thanks, everybody. So see you guys in 2 weeks. Bye-bye.
Bye-bye.
Thank you, José.